Hello, good evening. Welcome to the session. I hope I'm audible.
Good evening.
Yeah. I can see one person with raised hand. Do you have any query? Okay. If there is any questions, you can just unmute yourself or post to the chat box. Okay. Welcome to the second week live practice session for the NPTEL course Applied Environmental Microbiology. Uh, so we'll just start with the session now. I'll share my screen first. Okay. I hope the screen is visible now. So yes, we are in the second live session for this course today, and thank you for joining in. Uh, so, in the lecture videos that you would have gone through already, so the second week course content is about genetic codes, the translation process, about amino acids and protein structure, some basic biochemistry, followed by microbial growth curve, cell count, and then followed by some lectures on energy storage and the reactions that happen, basically the metabolic reactions that happen in the microbial cell. And then we have about microbial nutrition and fermentation and respiration. So today also we'll discuss like we did in the last class with some question and answer. I just post some questions for you to answer. You can just keep the answers in the chat box, and then we'll discuss about it. If you have any doubts from any of the lectures or a previous class, you can just post in the chat box, or you can unmute yourself and ask. So yes, we'll get started for today. Mm, okay, this is the first question for today. You can just read the question carefully and then try to answer with the option in the chat box. I'll give you a couple of minutes to solve each question. Okay, so the we'll just see the answer for this question. So as mentioned, it's both B and C. Uh, so all enzymes are 3D structures of linked amino acid. Yes, like mostly enzymes are proteins, so they are 3D structures of linked amino acids. And any given protein will have primary, secondary, tertiary, and quaternary structure that will give it uh, distinct properties. No quaternary structure is kind of not present in all proteins. Up till tertiary structure, yes, proteins do have primary, secondary, and tertiary structure, but the quaternary structure is uh, optional. And alpha helices and beta strands, they form the secondary structure in protein. And enzymes are biocatalysts and they are mostly proteins. Yes, that is also true statement. So yes. Okay, I have not highlighted B here. Before I upload the PPT, I'll do that. So it's both B and C. Okay, so the next question is based on the amino acids, on their classification. So based on the type of amino acid, three pairs are right and one is wrong. You have to identify which pair is wrong. Others, are you able to follow, or I am only getting response from one person?
Good evening, ma'am.
Good evening. Yes.
Ma'am, your voice is not coming.
Actually, I'm not speaking now. I've just given some time for you to solve the question that's displayed,
Okay, okay.
and then you
That's
can
great.
discuss.
Thank you.
Yeah. Okay. So the answer for this question is again B and D. So tryptophan is an aromatic amino acid. It has indole group, and then aspartate is not a positively charged amino acid. It is a acidic thing. So it is a negatively charged amino acid because it has CO minus group, because it is a acidic amino acid. And yes, cysteine is a sulfur containing amino acid, whereas tyrosine has phenolic group but no indole ring. So even that pair is wrong. So here aspartate is a negatively charged amino acid and tyrosine does not have any indole ring. So that is the answer for the second question. So now we'll move to the third question. So again, you have to identify which of the following statements is false. Whether the statements are that the tryptophan is coded only by one codon, stop codon is also known as nonsense codon, start codon codes for methionine, and Vobel base pairing amplifies the effects of DNA mutations. Which of the statements are false? Okay, we'll just discuss the answer for this question. So tryptophan is coded only by one codon actually, and it is UGG. So for other amino acids, you can see that there can be more than one codon that has coded for the same amino acid. So the first statement is true. And then the stop codon is also known as the nonsense codon. Yes, because the stop codon doesn't code for uh, any amino acid, and it'll allow for the termination of the translation process. And the third statement is that we have the start codon, which is AUG. So this codes for the amino acid methionine in bacteria and archaea, both the systems. And the last thing is about the Vobel base pairing. So Vobel base pairing amplifies the effects of DNA mutation. That is the statement. So first of all, if we see what is Vobel base pairing, uh, like when we are doing this transcription, translation and all, there is going to be a codon and there is going to be a tRNA that's going to bring the amino acid corresponding to the code. So sometimes what happens is there is less specificity between this codon and RNA matching, tRNA amino acid matching. Okay, so like especially with respect to the third spot in that codon. So what happens is, even though when the third spot is going to be different, still the same amino acid comes and attaches. So this is known as Vobel base pairing. And this Vobel base pairing actually does not amplify the effect of DNA mutation but minimizes it. Because what is DNA mutation? DNA mutation is the change in the sequence, like DNA sequence, right? If there is going to be a change in one particular place, then it means that the amino acid sequence could be changed, and therefore the protein structure and hence the functionality itself is going to change. So that is how mutation is going to adversely affect any organism. But when we go for Vobel base pairing, it is less specific, especially with the third position of that codon. So there are chances that though the uh, codon has one position swapped or changed with any other nucleotide, but still the correct amino acid comes and sits in the DNA sequence. So in that case, what happens is Vobel base pairing will minimize the effect of DNA mutation, and henceforth only that statement is the false statement here. The other three statements are true. So with that, we have seen about amino acids, codons, protein structures, and all. So as you can see this image, it talks about the four different structures of a protein: primary, secondary, tertiary and quaternary. So in primary, it's just a chain of amino acid, and this chain is usually connected through peptide bond, right? And so we call it a polypeptide chain. So there is going to be one amino acid which has amine group as well as the acid COH group and some functional group as well. Okay, so this is the primary structure. You can see in farther right that how the peptide bond is formed. So you can see that the OH group from the carboxylic acid functional group in one, and the hydrogen from the ammonia in the next amino acid is going to form water, hence forming a bond between this carbon and nitrogen, which is known as the peptide bond. So similarly, when different amino acids are linked together in a stretch in a chain, it is called a polypeptide chain, and this is the primary structure of the protein. So in secondary structure, you can just generally classify it as alpha helix and beta pleated sheet, and you have some bonding that occur between amino acids. So it gives some helical or sheet kind of a structure. And then based on the nature of amino acids that is present. Furthermore, bonding can happen apart from hydrogen bonding, van der Waals bonding. There can be some more kind of bonding also, which will lead to formation of 3D structures. So we had a chain, and this chain is now folded based on the chemical bonding that it can make with other amino acids, and this will confer it kind of a 3D structure. And finally, quaternary structure is not present in all the proteins, but there are proteins like when two or more polypeptide chains come together and they share some chemical or physical bonds, uh, they form a quaternary structure. As you can see, it needs two subunits, at least two subunits, subunit 1 and 2, and they are bonded with some chemical bonding. So these are the four structures that you can have for a protein. And the next one is the amino acid structures. So in this amino acid structure, you should just have in mind what are the classifications that are possible. For example, acidic amino acids, which is aspartic acid and glutamic acid, and basic amino acids like histidine, lysine, and arginine. So this is very easy to remember. Okay, acidic and basic amino acids. Apart from that, you have to know aromatic amino acids as well. And this classification, amine group containing amino acids, all of it, if you know, like it will be better to answer questions. So you have to know the names and kind of the classification of each of the amino acids. And then this table shows the genetic code table. So we all know that we have a DNA sequence which will be transcribed to RNA, and then there will be translation process for the protein formation. So in protein, for until DNA and RNA, you have this nucleotides, whereas in protein formation, it's about amino acids. So each three nucleotides in the DNA and RNA is going to correspond to one um, amino acid. So whenever in the translation process a group of three amino acids known as a triplet codon are going to be detected, the corresponding amino acid will be carried by a tRNA. As you can see this structure in the other side of the image, bottom right, you can see that this is a tRNA. So tRNA again will have the anticodon in it, and it will also carry the amino acid with it. So whenever this the codon or the in the mRNA and this anticodon are like complementary, they bond and the amino acid is now added to the protein chain. So that is how the translation process work. In this only, there is this concept of Wobble base pairing. Uh, so you can see here GGU and GGC is present, but CCG can is the tRNA which is having an anticodon called CCG can come and pair with both of this, giving glycine in that position. So this amount of lesser specificity with respect to the third position in the translation process leads to verbal base pairing. So that is what we saw in the last question. So these are the basics about amino acids and proteins. I hope it is clear. If there are any doubts, please post in the chat box. So the next question is about the bacterial growth. So next few questions will be about bacterial growth, and then we can discuss on it. So the first question is, in logistic growth model, the nutrient limitation plays an important role in determining the final population. Whether the statement is true or false.
And the answer is true.
Okay. Okay, so as told, the answer is true. So, in order to understand why the answer is true, we have to just see about what is the logistic growth model. So, in logistic growth model, you're going to have a equation, a mathematical equation, which states that dn by dt is equal to rn into 1 minus n by k, right? So, in this, n is the population size at any given time. So, and we have three factors in this equation. One is n, one is r, and another one is k. So n is the population size, as we know, and r is the uh, growth rate. Okay, and then k is the carrying capacity. So based on this equation, we can see that the growth rate is related to the the number of cells, or the final population is related to the carrying capacity. Okay, so carrying capacity, in the sense, it is the uh, maximum population that the environment can support. So the environment, in the sense, it depends on the nutrition that is present. Initially, when you are going to inoculate any substrate, you will be inoculating with uh, a representative small amount of microorganism. So at that time, there are going to be very less microbes, and there's going to be very high substrate. This means the resources are going to be plenty, and once our microorganism is adapted to that situation, then it starts growing like in a exponential manner. But once the cells grows in number and in size, the nutrient is going to be limited. So that time, only how much the system can carry, that much growth only will occur. So in that case, in logistic growth model, nutrient limitation is very important to determine the final population. So if you have some 100 milligram of some substrate compared to 200 milligram of substrate, there is going to be a difference in growth of that microorganism. So that is what the statement explains, and this we are relating it to the number of cells related to carrying capacity in the logistic growth model, and the carrying capacity in itself means about the environment, including the nutrients that are present there. So that is why this statement is true. Again, we have four statements. You have to identify the true statement about growth. So you all know that when we have a broth, we try to grow cells. We use the spectrophotometric analysis to just see, quantify growth basically, and we can visibly see turbidity. Just if you use a nutrient broth, the nutrient broth is going to be a clear liquid. Once you have inoculated and there is enough growth, there will be high turbidity in it. So statements are based on the turbidity measurement and growth of microbes.
Ma'am, screen is not visible.
Is it same with everybody?
I'm not able to see the slide.
Is it same for everybody? Can someone just respond in the chat at least? I have just reshared it. I've stopped sharing and reshared it again. Hope it is visible now.
Yeah, ma'am. Now it is visible.
Okay,
Thank you.
it's okay. So yes, we'll see the answer for this question now. Low turbidity signifies exponential phase in logistic growth model. Uh, so this statement has multiple aspects that we have to see now. So one is about the logistic growth model that we saw in the previous question itself. So in logistic growth model, we are going to talk about a logarithmic growth or a exponential growth. Obviously, that is what is logistic growth model. And as I explained just a few minutes back, we are going to measure the microbial growth based on turbidity. So as I told you, in very simpler or general context, let's take some nutrient broth. So we'll prepare the nutrient broth by adding the uh, media into some water and then autoclave it. So once we autoclave, we can see that the liquid is going to be very clear. So to that liquid, we are going to inoculate a little amount of a microorganism, just a few microliters of our microorganism, and then it starts growing. So this clear liquid then starts becoming turbid because there is cell growth in it. So as the cell is going, or the microbe is going to grow in that media, there is going to be higher turbidity. The turbidity is going to be significantly high. So in exponential phase, there is going to be exponential growth of the microorganisms, which means the growth is very rapid. Therefore, if there are more number of microorganisms, the turbidity will be high. So this statement is false. If it says that high turbidity signifies exponential phase in logistic growth model, then the statement can stand true. And then coming to the next statement, which talks about measurement of turbidity and plate count are direct methods for microbial cell measurement. No, uh, turbidity is an indirect measure. The plate count process is a direct method, whereas the turbidity is not a direct measure of microbial cell growth. It's an indirect method. And the third statement is the extent of lag phase depends on the age of the inoculum. This statement is the true statement that is given here. So this is because let's say we have an inoculum. So where will we get the inoculum from? It is an already existing microbe or bacteria that we have. So we might have gotten it from any stage of its growth previously. Let's say it was in lag phase, log phase, or stationary phase. Uh, so this will decide the age of the inoculum, right? So if it is an exponential phase, then maybe it can come here. Like we can say that the inoculum is a little bit fresh, so that its adaptation time will be lesser. But if the bacteria is already in some stationary phase, then it's going to be a very aged inoculum, and hence its capacity to adapt to this new media or new environment that you're going to put in is going to be very low. Therefore, the extent of lag phase can be very high. It can either grow or not grow, that is a different case, but it will take more time to get adapted to because it's a old inoculum. So the extent of lag phase depends surely on the age of the inoculum. And the last statement is when the microbial population is lesser than the carrying capacity, microbial death starts to occur. So in the previous question itself, we saw what is carrying capacity. Carrying capacity is decided by the environment based on the nutrient availability and the environmental condition, how much bacterial growth can be supported. That is what is carrying capacity. So only when you reach the carrying capacity, then there is this discussion about depletion of this nutrients, or the nutrients becoming limited. So therefore the microbial population, if the microbial population is lesser, then the death doesn't start to occur. Once it reaches and exceeds this carrying capacity, then only death will start to occur. So that is why the last statement is also a false statement. So the answer for this question is again C.
Ma'am, third, please, just third point.
Sorry, which?
You can please explain again third point. The effect of stationary, if your inoculum is in stationary culture, then lag phase is more or less.
No, if the inoculum age is more, then the extent to which it can adapt will be lesser. So it can't adapt very rapidly to a new environment. So in that case, when we are going to use the aged inoculum, then the lag phase is going to be prolonged. Whereas when we use a inoculum which is way younger, then the lag phase is going to be shortened. So this way there is a correlation between the extent of lag phase and the age of inoculum that we are using it. So
Okay, thank you.
yeah, okay. I hope it was clear. And now we go to the next question. So the decay term in the growth model is important because of which of the following? Okay, so the answer for this question is again B. Uh, so when we talk about microbial growth model, there are we have this logistic or monod model, and in a typical growth curve, you have different phases like a lag phase, log phase, stationary phase, and the death phase. So this death phase is what gives this term called a decay term. And so in the stationary phase, what happens is we experience some kind of nutrient depletion. So the rate at which the cell growth and the rate at which it decays is going to be uh, like similar. So that is why you have a very stationary phase there. It's not showing any elevation in terms of the curve, it's just going to be flat. But when we come to this death phase or decay phase, here the nutrients are completely depleted, there is accumulation of the waste product, all of it is happening here, and hence the microbes are no more able to survive in the environment, and the number of viable microbes is going to decline, and the microbes are going to die. So the term that incorporates this factor in our growth model is known as the uh, decay factor. So yeah, it makes the model more sensible, but the real reason is it incorporates the removal of microorganisms due to natural death. So we also have to account that when we are modeling the growth of microbes. Another question, just a basic question: why are semi-logarithmic plots preferred over arithmetic plots for analyzing microbial growth kinetics? So yes, the answer for this question is that the semi-logarithmic plots linearize exponential growth data. So when we are going to plot our microbial growth curve, we are not going to plot the direct arithmetic data in the uh, graph sheet as a arithmetic plot. Instead, the y-axis, what we do is we try to take logarithm, and then the x-axis is going to be time. So this is how our microbial growth curve is going to be plotted. So when we are why are we linearizing? Because when we are going to directly plot the exponential growth data in a graph sheet, what happens is you get a very steep curve because you're not doing any kind of transformations there. So it's going to be a very steep curve, and it's difficult to interpret. And one more thing is there is going to be exponential growth, and manually plotting that data in arithmetic plot is going to be very hectic, and to derive interpretation from that is also going to be very tedious. And henceforth, when you are going to do some transformations like bringing it to a logarithmic scale, what happens is you can accommodate like several orders of magnitude of growth in that graph itself. And when you have this linearized version, you just have a line that specifies a particular phase, and those lines will have a particular slope, and that slope is what is we call as the rate of the microbial growth. So when we are going to linearize the data, what happens is we get some lines instead of some steep curves, and these lines are going to hold a slope which corresponds to the growth rate at that particular phase. So that is why we use semi logarithmic plots to display the microbial growth kinetics. So it does not show some nutrient limitations, or it doesn't eliminate lag phase. It's just linearizes the exponential growth data and becomes very obvious for interpretation and analysis. Okay, so the next question is, if two cultures have identical optical density in the sense like we talked about turbidity, absorbance value in one of the questions. So when we are going to have some two cultures that are having similar optical density, but they are in different growth phase, which statement is the most accurate among the following? So this is just a case-based question. You have two cultures. You are just going to find the optical density of both the culture, and that is found to be very identical. But the two cultures are not in the same growth phase. Which statement is the most accurate? for this question is again C. So why? Because we saw that this turbidity is kind of a indirect cell measurement technique, right? So let's say we have two cultures and they have the uh, same optical density. So one culture can be in exponential phase and the other culture can be in a stationary phase. So we don't know if both are going to be in the same phase itself. So hence their metabolic activity is, we cannot conclude that their metabolic identity activity is identical, because their metabolic activity depends on how fast they can metabolize the substrate and grow, and it's mentioned that they are already in different growth phases. So uh, they are not metabolically identical in terms of activity. And then we are not doing any cell size measurement when we are talking about optical density. So again, that is wrong, but it states that population level measurements mask individual cell differences. That statement is true. And coming to the last statement first, like they have the same generation time, no, that we can't conclude. And generation time, mostly we can compare only with in terms of exponential phase, right? And we don't even know that if we have the data in the question, it is not mentioned whether we have the full data of when the exponential phase is starting, when it is ending, what's happening in that. So only when we have that data we can talk about generation time. So here talking about generation time also makes no sense. And but when we talk about population level measurements, uh, one more thing that we have to account is in terms of optical density measurement, we are not going to see whether the cell is a live cell or a dead cell, which means a viable or a non-viable cell. Uh, all are going to be measured, because the OD is about if the cell is present in the liquid or in the sample or not, not about like whether the cell is viable or non-viable. So in that case, this population level measurement is going to mask the individual cell uh, differences when we have identical optical density but difference in their growth phases. So C is the most appropriate option for this question. So yes, with that, the next question is, in a chemostat or a continuous reactor, increasing the dilution rate beyond the critical value will result in which of the following? This is a very direct question. reactor, and this we use for continuous microbial cultures. So we have batch process, we have semi batch process, and continuous process. So in batch process, what we do is we just have substrate in a reactor, and we add the inoculum to it, and we allow it to grow and produce some product that we intend to produce. In this, what you do is you neither add any more substrate, you neither take out anything out from any product or that mixture out from the reactor until the completion of the reaction. So if your reaction time is, let's say, some 5 days, uh, at day 0 you're going to add your substrate in inoculum, and at day 5 you're going to terminate the reaction and then take out the product. Whereas in case of fed batch or semi batch processes, what we do is we have a reactor and we intermittently feed the reactor, let's say a period of 5 days, and after 2.5 days we just feed the reactor once, or every day we are going to feed some amount of substrate into the reactor. So that, but we are not going to take anything out of the system again, and that is a semi batch process. But in a continuous process, what we do is we keep on adding the nutrients or the substrate into the reactor, and simultaneously an equal amount of the product or the reaction mixture is going to be taken out of the system as well. So this is a continuous process, and here when we are going to add this nutrient and maintain it in a steady state, we call it as a chemostat process. So we are going to maintain the uh, concentration of the nutrient or the substrate constant in the inside the reactor by continuously adding new substrate and removing the utilized substrate and the products as well. So this is the principle of chemostat. So based on this, we have something called a dilution rate. So when we are going to add a new set of substrates and remove the already existing substrate, uh, the microbial population inside is going to get diluted. uh, so there is a rate at which we have to do this, a rate at which, or should be some fixed interval in which we are going to add the new substrate and remove the utilized one. Okay, and this rate should correspond to the growth rate of the microorganism. So let's say a microbe uh, grows has a growth rate which corresponds to some amount of its retention inside the reactor. If we are going to add the substrate in a faster pace than this, then what happens is before even the microorganism can grow properly, we are trying to take it out of the system. So the amount of microorganism present in the inside the system is going to be reduced in that case, which we technically term as washout of microorganisms. Let's say we have the dilution rate and this growth rate like compatible to each other, then there will be a steady growth of the microorganism. So even if you remove some amount of the mixture outside, still you will have enough microorganisms to continue the process to utilize the substrate uh, that is being freshly added into the system. So in that case, in chemostat, increasing the dilution rate beyond the critical value, there is a value called a critical value, which again is related to the growth rate of the microorganism. If we are going to exceed beyond this, then the viable cells are going to be washed out of the system, and there is going to be washout of microorganism, and that is the correct option here. So with that, we have seen a few questions about microbial growth and chemostat and stuff. So as you can see, the left the picture shows you the a typical growth curve for a microorganism consisting of all the 4 phases. And on the other side, you can see microbial cell counting technique. We saw about direct and indirect techniques. So direct techniques are plate count and cell counter techniques, whereas indirect, you measure the turbidity, metabolic activity, and dry weight. Like they are all indirect methods of microbial cell counting. You just assume that if there is this turbidity or this absorbance value, then this amount of growth could have happened. So you indirectly correlate, you don't directly count the number of cells in them. Whereas in plate count method, you're going to count the cells that are growing in your plates. So yes, with that we'll move to the next topic. So this is again based on microbial growth and the kinetics part only. This is just one numerical question to solve. I think I have one or two questions only in the entire presentation. So this is the first question. If one starts with 10 to the power 4 cells, the number of cells is 10 to the power 4 in a culture, and the generation time of that culture is going to be 2 hours. How many cells will be in the culture after 48 hours? I think it's a very simple question. You can just directly use the formula and then get the answer for it. You can just post the answer in the chat box. So you're going to start with 10 to the power 4 cells, and the generation time of those cells is going to be 2 hours. At the end of 48 hours, how many cells will be there in the system? answer counts up to 1.7 into 10 to the power 10 cells, if I'm not wrong. So here goes the answer. The formula that you have to use here is x is equal to 2 power n into x naught, where x is the number of cell after n generations, which is what you have to find here. And you know that you have to find the number of generations as well, right? So in order to find that data, you have the generation time and the time for which your reaction or your experiment is going to run for. So you're going to run the experiment for 48 hours and the generation time is 2 hours. So in that case, there is a possibility of 24 generations that will occur during the given time. So here the number of generations is 24, and 2 power 24 into x naught is the x naught is the initial number of cells, which is 10 to the power 4 cells. So 2 power 24 into 10 to the power 4 is going to give you 1.7 into 10 to the power 11 cells. I hope the answer was clear. Yeah, it's approximately 1.7. That is fine. Just check with the decimal. I'm just worried. Sorry, check with the exponent. Yours counts up to 1.7 into 10 to the power 11 or 10. That's my question. The answer is 1.7 into 10 to the power 11. So it'll be easy if you can practice giving the answers in terms of exponents only, so that you don't miss out any digit, and that will make a huge difference in your answer. Okay, so with that, okay, yeah. This is about the Gibbs free energy. Yes, as you have mentioned, this is just a direct formula question. So general equation about the Gibbs free energy change is that del G is equal to del G naught plus RT ln K. So here you might be confused with the last one also. The difference is that there is no minus sign here. So when you talk about del G being 0, then you get this, and you also don't have a logarithmic scale there. So if you have a confusion, then maybe I tried to explain that, but this is just a direct question about some equation. Okay, now again about the same Gibbs free energy. Why is del G, which is the actual free energy change, more biologically relevant than del G naught prime? Try to understand the difference between del G and del G naught prime and then answer the question. So both del G and del G naught prime are going to talk about the Gibbs free energy or the standard free energy change only. But in case of the del G naught prime, what happens is it is the standard free energy change at specified conditions of 1 molar concentration of the reactants and products, 1 atmosphere pressure, and 298 Kelvin temperature. So this is the standard reference point at which you have to calculate your del G naught prime. But when you talk about del G, uh, this doesn't follow any standard condition. It just goes with the condition where the cell is present only. So in when we talk about biological system, we are not going to have biological reactions being happening only at the standard conditions, especially the condition of having 1 molar concentration of the reactants and products is not going to work out with the biological system itself. So in that case, del G accounts for the intracellular metabolite concentration, which is generally very less than 1 molar concentration assumed by the calculation of del G naught prime, and hence del G is more biologically relevant than del G naught prime usage. It has nothing to do with temperature or the kind of metabolism that's happening, and it's not also constant for all reactions. Okay, again, one more easy question, I guess. Most energy rich compounds in cell primarily are talk about energy rich compounds, the immediate compound that comes to our mind is ATP. So it's very obvious that phosphorylated compounds with ester or anhydride bonds are mostly energy rich compounds, and the classic example is going to be our ATP. When we go to the other options, mostly energy rich compounds are broken down for energy release. So they undergo exothermic reactions and not endothermic. They don't need any energy to be broken down. And they are also usually larger in size. Like when we talk about adenosine triphosphate, it is the adenosine group with three phosphate groups attached. So it's going to be a larger molecule and it does not pass through membranes that easily. And also, the speed of replication has nothing to do with a compound being energy rich or not. So in that case, most energy rich compounds are phosphorylated compounds, and the classic example to remember this is ATP. Okay. The next question is, which kind of the reaction has the most energy available for growth and replication? Right, again, aerobic oxidation. So here we have oxygen that is used to break down the organic matter completely. So, and when you track the glycolysis process and the amount of ATP generated in this kind of metabolism, you can see that the aerobic process, because of use of oxygen to break down the organic molecules, you are going to get around 32 ATPs. Minimum of 32 ATPs will be generated by this oxidation or this metabolism. Whereas when you talk about anaerobic metabolism and all, there is not going to be much oxygen, and the ATP yield is going to be lesser in those cases. So aerobic oxidation has the most energy available. You can just simply remember the number of ATPs that are produced, based on which you can answer this question. Again, which of the following is not a long-term energy storage compound? If you know about the microbial metabolism well, then this question can be answered so quickly. glycolysis pathway, you have glucose, you have then glucose 6 phosphate formed there. So it's not a very long-lived compound. It's just the intermediate in the metabolic process. Whereas um, PHAs and PHBs, maybe in the short form, you might have heard about these terms. Glycogen, yeah, it's a long-term energy storage compound, obviously. And PHAs and PHBs, maybe you might have heard it in that way, where microbial cell tries to produce these polymers, uh, which is a long-term energy storage compound, and when in terms of need, it again breaks down this compound for energy if there is some energy shortage. So this PHB accumulation occurs at a particular condition, and this PHB again is a industrially relevant compound only. Many people are working on the production of PHBs. So based on that, you have to maintain certain conditions so that your cell goes and produce this PHBs actually. So if you're aware at which condition these can be produced. talk about this oxygen absence, uh, this is not very predominant. Oxygen being absent is not like the most likely scenario where PHB is being produced. And finally, PHB is in itself is a energy storage component. So when the ATP demand is going to be very high, a cell will not produce a energy storage component. Rather, whatever the energy storage component is already produced, maybe it could be broken down because the ATP demand is high in the system. So in that way, even that is the wrong option. The most likely condition for PHP production and accumulation is nitrogen should be limiting, but the carbon should be excess, so that the cell tries to store that carbon by the production of PHP. Okay, one more question to understand the chemistry of this energy rich compounds. So yes, here when we talk about anhydride bond and ester bond in phosphorylated compounds, so phosphorylated compounds are going to be AMP, ATP, and all those stuff. So when we talk about this anhydride bond, it's mostly present in molecules like ATP. So phosphorylated compounds, as we saw, we are going to use them for this energy storage, right? Like we store energy in the form of ATP, and we again break down the ATP to release the energy and utilize the energy for other metabolic reactions. So it's kind of a trade-off for energy. In that case, the bonding plays a very critical role. Which bond, while broken, gives you the maximum energy? That will be most favorable, because you can store a lot of energy in that way, right? So the distinguishing feature between anhydride and ester bonds in phosphorylated compounds is going to be the high free energy of hydrolysis. Whenever you hydrolyze the compound, whichever is giving you the highest energy, you'll be concerned about it. And yeah, they are present in energy transfer, and the molecular weight or the presence of nitrogen is not very important in this context. The most important thing in phosphorylated compounds is their association with energy storage, and for that you are very concerned about the high free energy of hydrolysis. Okay. And the next question is, identify the true statements from the following. This is based on the electron transfer. obviously, because the flow is going to be very spontaneous, as it is was mentioned in the previous statement. So in that case, you will have a free energy release there. You don't need any energy for that to happen. And then ATP is formed by phosphorylation reaction. Obviously, yes, you need three phosphate molecules to get attached to adenosine in order to form a ATP. Therefore, it's a phosphorylation reaction. And finally, NADH is a coenzyme. So you usually represent these as NAD plus NADH combination. In that case, you call the entire combination as coenzyme and not only one component of it. And based on this only, I tried calling NADH also as a coenzyme. So with that, we just sum up whatever we saw. And in order to talk about all these things and about the phosphate bonding, how much energy the bond holds, how much free energy the bond holds, all those stuff is present here. The important thing to note here is the one in the bottom left corner, which is the uh, electron tower. So in this electron tower, the electrons will spontaneously move from donors to the acceptors, and donors are generally present higher on the tower, and they have more negative potential, whereas the acceptors are going to be present on the lower end of the towers with more positive potential. So there is a spontaneous electron transfer from the top to the bottom of the tower, and depending on the distance that you can cover in this tower, you can see the energy release. So you keep this concept in mind. In future sessions also, we'll be using this concept when we talk in detail about the aerobic anaerobic processes and all. So because in aerobic and anaerobic processes, your terminal electron acceptors are going to be different. So based on the terminal electron acceptors, how this energy change or the difference in the amount of energy released is present, all that we'll be seeing. We also saw one more statement previously in one question about this aerobic oxidation, that it generates around 32 ATPs. All these concepts are interrelated, and in one of the future lectures we'll be seeing in detail about this as well. So for now, you have to know that there is a concept called electron tower, in which there is spontaneous flow of electrons from the top to the bottom, and the top is going to be uh, good electron donors with high negative potential, whereas the bottom of the tower is going to be acceptors with relatively positive potential. And you can also refer about the different oxidation processes here in the right. Again, a very simple question: which of the following is a micronutrient? Right answer is copper is a micronutrient. Sulfur, phosphorus, and nitrogen, we need more of them in cells. So it's not a micronutrient, it's beyond that. And yeah, now we have the next question, which is identify the false statement. We just have a few questions to go. We'll just see one by one these. And this part is going to be about the microbial nutrition. A few questions on microbial nutrition we are going to see. to harvest the light energy using the oxygen, that is correct. And then chemotrophs do not utilize light as energy source, yes, because they don't perform any photosynthesis or something, but instead they use chemical energy, they don't use any light energy. And finally, coming to the statement where carbon dioxide is the energy source for chemoautotrophs, it is not the energy source. uh, Carbon dioxide is not the energy source in chemoautotrophs. They just derive their energy from inorganic substance, not carbon dioxide. Whereas carbon dioxide is only the carbon portion of the energy, and that is why it is the false statement. The next question is a microorganism using organic carbon as both energy and carbon source is called. So the questions in this section is kind of repetitive only, just to make your mind clear by repeatedly solving the same concept, so you don't get confused within the terms. You have chemo, photo, auto, hetero, all these terms in different combinations, including locally. So you should not get confused when you are approaching the questions, and hence I have a set of questions in a similar pattern. Right. So you when you talk about organic carbon as energy source, and carbon source is going to be chemoorganotrophs. So you have organic molecule serving as the energy source and as the carbon source. The next question is which condition favors fermentation over respiration? Exactly. So when you have the absence of external electron acceptors, instead of respiration, fermentation process is allowed. And as a continuation of discussing this question, maybe in the next session, the first question that I'll be having is what the difference between fermentation and respiration. If we have time, we'll just discuss. We'll just see through the questions first, and if we have time, we'll discuss, or in the next class, we'll discuss. Again, another question related to the same concept. Compared to respiration, fermentation generally yields. Exactly. Compared to respiration, fermentation generally yields lesser ATP but faster regeneration of NAD+. So you can see that we have an absence of external electron acceptor, and we have less ATP generation in the process. I think this is the final question. It is which conclusion best integrates microbial energetics and environmental engineering? Uh, in view of time, I'll just show the answer and discuss them, and before you could answer. So the statements are maximum growth rate always maximizes treatment efficiency. Not exactly, because even if you have some maximum growth rate, it's about how metabolically the treatment is feasible. So microbes can grow, but metabolically how much the treatment is feasible depends on what target component you want to eliminate from the system. So all of it matters. So maximum growth rate does not always maximize treatment efficiency. And bioenergetics is irrelevant at reactor scale. No, it is very much relevant. Only based on bioenergetics we saw a concept of critical dilution factor and washout. And when you even model, you have this growth rate taken into account. All of it is a part of bioenergetics, so it is relevant at the reactor scale. And maintenance energy is negligible. No, it is very critical, because first the cell has to survive, then only it can do any other function that you want it to do. So maintenance energy means the housekeeping energy for the cell to basically do its fulfill its basic metabolic needs for survival, and then only we can go about all the treatment processes that we intend to do. So in that case, maintenance energy cannot be very negligible. But energy conservation strategies influence reactor design. That is true. So if you can change some temperature, pH, or the nutrient condition, or you can supply with any other factor that can affect your microbial growth. All these are energy conservation techniques, and this surely influences the reactor
Break
design.
hold for
So energy, if you're going to conserve energy, then that energy can be used for your treatment process, and the size of your reactor can be reduced when you're going to model any reactor or something. So in that way, environmental engineering is totally about using the microorganisms for detection or treatment in the environmental system. So these are the two major applications of microbes in environmental engineering. In that case, your energy conservation strategies will influence your reactor design. So yes, this chart typically summarizes uh, the different carbon source and energy source that your organisms can have, and also this is one kind of a color periodic table which shows you about the nutrient requirements of the elements in microbial group. You all of you would have known about the periodic table classification, but you just be aware about the nutrient classification, um, essential nutrients, micronutrients, macronutrients, all of it. So yes, this is the final question. Which of the following is attributed to fermentation? So the answer is 1, 2, and 4. 1, 2, and 4 is glycolysis. Yeah, you have glycolysis. And fermentation route, there is a shift to the fermentation route when there is some kind of anaerobic condition that is found. And in fermentation, you have substrate level phosphorylation, which is a direct kind of a phosphorylation of the substrate. But in fermentation process, you will not be hearing about this proton motive force and the oxidative phosphorylation. So before starting off the next session, I will talk in detail about the difference between this respiration and fermentation, and in that we will see about what is this substrate level phosphorylation and oxidative phosphorylation and stuff. I hope that is clear. So with that, if you have any particular doubts about any questions or concepts, you can just post in the chat box, or else thank you all for your presence and listening, as well as your active participation. Hope to see you all in the next session as well. Thank you for joining in. If you have any suggestions or comments, also please post in the chat box so that I'll prepare the next session accordingly. I hope this way of question and answer session is fine. Thank you so much.